For decades, an untreatable diagnosis was just, well, it was like a heavy lock steel door.
Right.
Doctors would basically just stand in front of that door, look at patients and their families, and, have to deliver the absolute worst news imaginable.
Yeah, just simply saying they couldn't get through.
Exactly. It was binary, it was heartbreaking, and it carried this terrible, absolute finality to it. Welcome to today's deep dive, by the way.
Thanks. And, we really like to think of modern medicine as omnipotent, right? Like we can fix anything.
Right, for sure.
But the reality is the medical landscape is still full of those locked doors. The barrier might be a tiny mutation in a single strand of DNA or a missing protein, or honestly, just the physical impossibility of operating on a really fragile, failing organ.
Yeah, but today we're not just staring at the door anymore.
Yeah.
We are looking at the people who are like building the master keys.
Oh, absolutely. Taking a sledgehammer to the word untreatable.
That is the mission for this deep dive. We are unpacking 3 incredibly exciting clinical trial summaries that are actively redefining that word. We're looking at major breakthroughs in really how we deliver a cure.
And the stack of sources you brought to the table today, I mean, it covers a remarkable spectrum. We're jumping from literally reprogramming the genetic code of young children all the way over to performing robotic heart surgery on adults.
Right. Which sounds like sci-fi, but despite how different these trials look on the surface, they all share an exceptionally high breakthrough potential score. We're talking 18 out of 20.
Which is huge.
Yeah, and their overall TrialsAlert scores are hovering right there in the high 70s. So whether you are a medical professional looking for the cutting edge or, you're just someone fascinated by human ingenuity, you're going to want to hear this.
It really shows how the most devastating prognoses are just being completely rewritten.
Okay, let's unpack this. I think the best place to start is at the absolute microscopic level, right?
Yeah, let's dive into the genetics. Specifically, we are looking at a profoundly difficult condition called X-linked myotubular myopathy, or XLMTM.
Okay, XLMTM.
Right. And to really understand the gravity of this trial, you have to understand the biological mechanics of the disease first. So, XLMTM is a rare genetic neuromuscular disorder, and it's caused by mutations in the MTM1 gene.
And from what I read, that specific gene is the one responsible for, normal skeletal muscle development. Right?
Exactly, which means boys born with this mutation, they completely lack a crucial protein called myotubularin.
Okay.
And without myotubularin, the muscle fibers simply do not structure themselves correctly. They remain incredibly, profoundly weak.
We aren't just talking about a mild delay in crawling or walking here.
No. We are talking about infants who, in most cases, lack the sheer muscular strength to even inflate their own lungs.
Oh, wow.
Yeah, they cannot breathe independently. They spend their lives entirely reliant on mechanical ventilators and feeding tubes. And historically, survival beyond early childhood is just exceptionally rare.
I mean, that is just an incredibly grim baseline. But this brings us to the intervention. So the trial data shows an international phase 2 study involving 27 children, and they're all under the age of 5.
Right.
The treatment is a one-time intravenous gene therapy. I'm looking at the drug name here. Resomurgine bilparvovec.
Yeah, which is quite a mouthful. It's often just referred to as AT132. And the mechanism of action here is really where the science fiction becomes reality.
Yeah, explain that, because it's wild.
So they are using an AAV8 vector to deliver the therapy. An AAV, which stands for adeno-associated virus, is essentially nature's most efficient infiltration machine.
Because viruses are literally built to break in.
Exactly. Viruses have evolved over millions of years to do one thing: perfectly breach the cell membrane and inject their genetic material into the nucleus.
So the scientists are essentially, hijacking that natural viral mechanism for a rescue mission.
That's a great way to put it. They completely hollow out the virus. They take out all the viral disease-causing DNA. They strip it down until it is nothing more than an empty microscopic delivery vehicle.
Just a shell.
Right. Then they take a perfectly functional, correct copy of that missing MTM1 gene and they load it right into the viral shell.
Wow.
And when they infuse this into the patient's bloodstream, trillions of these hollowed out viral vectors travel through the body. They latch on to the muscle cells and they inject the correct genetic sequence.
So the body finally receives the instructions to start producing myotubularin on its own. It's rebuilding the muscle structure from the inside out.
Exactly.
It's think of it like a massive manufacturing plant that is missing a single crucial blueprint. The heavy machinery is all there. The raw materials are piled up. The electricity is on. But the workers are just standing around because they don't know what to build.
I love that analogy.
Right. And we're basically sending in a massive fleet of delivery trucks, the AAVA vectors, to drop off the correct blueprints right onto the factory floor. And according to the initial data, the factory actually springs to life.
It really does.
Children who had never taken an independent breath in their lives were suddenly achieving ventilator independence.
Yeah, the improvements in motor function and respiratory independence were just paradigm shifting for a disease that previously offered zero hope.
But wait, I'm looking at this trial's data sheet, and it lists the status as active, not recruiting.
Yeah.
If these children are achieving something as miraculous as getting off a ventilator, why hit the pause button on enrolling new patients?
What's fascinating here is that you've hit on the most critical bottleneck in modern genetic medicine. The trial was paused for new patients because of serious hepatobiliary adverse events.
Hepatobiliary, so liver issues.
Yes. To understand why this happens, we have to look at how the body processes that massive fleet of delivery trucks you mentioned. When you inject trillions of viral vectors into a human being, they don't all magically make a beeline for the skeletal muscle.
Right, they're just in the blood.
Exactly. The blood circulates systemically, and a massive portion of that cardiac output goes straight to the liver.
Because the liver is the body's primary filtration plant.
Right. The liver is just packed with specialized immune cells whose entire job is to scan the blood for foreign invaders and clear them out. So suddenly, the liver is flooded with an astronomical number of viral particles.
Oh, I see. Even though the virus is hollowed out and totally harmless, the liver's immune system doesn't know that.
It has no idea. It just sees an invasion. So it triggers a massive systemic inflammatory response trying to clear what it thinks is an infection. This creates severe liver toxicity, which, in a medically fragile child can be life-threatening.
God, that is an excruciating clinical calculation. You have a baseline disease that is almost universally fatal, and a therapy that proves it can work miracles, but the delivery mechanism itself carries immense systemic risks.
It's incredibly tough. AT132 has actually become the bellwether for all pediatric AAV gene therapy. Everyone working in the rare disease space is actively watching how researchers navigate this.
Because whatever they figure out here applies everywhere else.
Exactly. The lessons being painfully learned right now about how a child's liver processes this influx of viral vectors that will shape the dosing strategies and safety protocols for an entire generation of rare disease therapies. They have to map the exact threshold where the vector can deliver the payload to the muscle without, triggering a catastrophic immunological alarm in the liver.
Well, so if the problem with the AAV8 vector is that these delivery trucks are getting jammed up in the liver and causing a massive systemic pileup, the obvious question is, how do we bypass the liver entirely? What if, instead of trying to deliver the raw genetic blueprints to the factory, we just manufacture the finished product in a lab and deliver it directly to the specific cells that need it?
That perfectly brings us to our second trial. Infantile-Onset Pompe disease, or IOPD. This is another devastating genetic disorder, specifically a glycogen storage disease. In Pompe disease, the body is missing an enzyme called acid alpha-glucosidase.
And without that enzyme, the cellular machinery just starts to break down.
Yes. So glycogen is simply stored energy. When a muscle needs energy, the acid alpha-glucosidase enzyme breaks the glycogen down into glucose.
Okay.
But without the enzyme, the glycogen just builds up inside the cell's lysosomes. It's like a wood chipper that jams, but the logs keep getting shoved into it anyway.
Yikes. That sounds destructive.
It is. The glycogen accumulates to toxic levels, physically destroying the muscle tissue. In infants, this manifests as progressive muscle weakness and hypertrophic cardiomyopathy.
Which means the heart muscle becomes abnormally thick and struggles to pump right.
Exactly. An untreated baby with IOPD faces a very similar tragic prognosis to XLMTM, rarely surviving their first year.
So looking at the trial data, we are looking at an international phase 3 trial taking place across 16 sites, studying 17 treatment-naive babies for up to 4 years. And the intervention here is an enzyme replacement therapy, or ERID, called Avalglucosidase Alfa.
Right.
But delivering enzymes isn't a completely new concept in medicine, is it?
No, not at all. Enzyme replacement therapies have been utilized for a while, but standard ERIDs face a massive biological hurdle, which is inefficiency.
How so?
Well, you can intravenously infuse a synthetic version of the missing enzyme into a patient's bloodstream, sure. But getting that enzyme to cross from the blood into the interior of the muscle cell is incredibly difficult. Muscle cells do not just passively absorb whatever is floating by.
So it just goes to waste.
A large percentage of standard urate simply gets filtered out by the body and excreted before it ever even reaches the target tissue.
Okay, here's where it gets really interesting. Because Avalglucosidase alpha is engineered with something called enhanced mannose 6-phosphate targeting.
Yes.
Going back to our delivery truck analogy, we are no longer just throwing thousands of packages into the bloodstream and hoping the muscle cells pick them up. This new IRD uses mannose 6-phosphate, like a highly specific tracking barcode printed right on the side of the package.
That is exactly how it works. The surface of a muscle cell is naturally covered in mannose 6-phosphate receptors. They act like specialized cellular loading docks.
Okay.
So by biochemically engineering the replacement enzyme to be exceptionally rich in mannose 6-phosphate, the drug effectively carries the exact molecular key required to open that specific loading dock.
Oh, wow.
Yeah, the receptors actively grab the enzyme right out of the bloodstream and pull it directly into the lysosome where it can finally start clearing out that toxic glycogen buildup.
The precision there is staggering. Instead of flooding the systemic circulation and risking all those unintended interactions, like we saw with the liver in the first trial, we are ensuring the package only gets opened by the cellular address that has the matching barcode scanner.
It really is a generational leap in the fundamental biochemistry of enzyme replacement therapy. I mean, this trial isn't just tweaking a dose. It's proving that we can actively outsmart the body's natural barriers to absorption.
Which is huge for these kids.
Massive. If this phase 3 trial confirms the profound improvements we've already seen in older patient cohorts, we are looking at a complete reset of the standard of care for Pompe disease. We're talking improved survival rates, drastically reduced reliance on invasive ventilation, and a preservation of motor function. It transforms the entire trajectory of the disease from a rapid decline to basically a highly managed condition.
That's incredible. So we've been talking about navigating these microscopic biological pathways, dodging the liver's immune response and hacking cellular receptors with biochemical barcodes. But sometimes the greatest barrier to delivering a cure isn't a complex immune response or a cellular membrane.
Right.
Sometimes the bottleneck is purely physical. It is the absolute limitation of human anatomy and just, well, the human hand. Let's zoom out to the macro mechanics of the human heart.
Yes, let's talk about the physical side. The perfect example of this physical bottleneck is severe tricuspid regurgitation, or TR.
Okay, TR.
The tricuspid valve separates the right atrium from the right ventricle. In TR, the leaflets of that valve fail to close tightly, meaning every single time the right ventricle squeezes to pump blood into the lungs, a significant amount of that blood leaks backward into the atria.
It's sloshing back where it came from.
Exactly. This causes fluid backup, severe swelling, and eventually right-sided heart failure. Millions of people suffer from this.
Now, the traditional fix for a failing valve is open heart surgery. But reading through the sources, it seems this particular condition is vastly undertreated because that traditional route is simply off the table for most patients.
It really is. By the time severe TR becomes highly symptomatic, the patients are usually older, physically frail, and dealing with compounding health issues. And the right side of the heart is incredibly delicate.
So surgery is just too much for them.
The trauma of cracking a patient's chest open, stopping the heart, placing them on a cardiopulmonary bypass machine, it just carries an unacceptably high mortality risk for this specific demographic. So for decades, cardiologists had to rely on simply prescribing heavy diuretics to manage the fluid buildup while the heart slowly failed.
So the alternative to open heart surgery is a transcatheter approach, basically threading a flexible tube through the blood vessels to reach the heart. Which brings us to the third trial. This is taking place at the Prince of Wales Hospital in Hong Kong.
Yes.
It's a technique study focused on robotic transjugular transcatheter tricuspid valve replacement. That is a lot of words.
It is, but it's a game changer. Manual transcatheter procedures have already revolutionized cardiology, but the tricuspid valve is notoriously difficult to reach. In a manual procedure, an interventional cardiologist makes an incision in the jugular vein in the patient's neck.
Right in the neck.
Yeah, and then they have to push a long, flexible wire down through the blood vessels, make a sharp, complex turn into the right atrium, and deploy an artificial valve perfectly into place.
I mean, that sounds like parallel parking a semi-truck down a twisting alleyway. You were asking a surgeon to thread a needle while riding a roller coaster. They have to navigate that wire through the chaotic currents of flowing blood inside a violently beating heart muscle, all while relying on fluoroscopy.
Which is incredibly hard to read.
Right. Fluoroscopy is essentially a delayed, grainy, two-dimensional continuous X-ray screen.
And the physical toll on the surgical team doing this is immense because fluoroscopy uses constant X-ray radiation to visualize the catheter inside the body. The doctors have to wear heavy lead aprons.
Oh, those are so heavy.
They can weigh 20 pounds. And they are sometimes standing over the operating table for hours. The operator fatigue is just a massive variable in these surgeries.
But the introduction of a robotic system completely shifts the paradigm here. The robot provides stabilized, microscopic precision. It acts as the ultimate power steering and 3D mapping system, right? Eliminating the tiny hand tremors and the sheer physical exhaustion of fighting the catheter's tension.
Exactly.
But wait, I'm looking at the scope of this trial. The previous trials we discussed were global, multi-site efforts involving dozens of patients. This trial in Hong Kong is enrolling exactly 10 patients at a single center.
Right, it's very small.
I mean, if this robotic assist is the ultimate game changer for a disease affecting billions of people, why is the scope so incredibly small?
If we connect this to the bigger picture, the scale of the trial perfectly reflects where we are in the timeline of surgical innovation. This is a first-in-human technique study.
Oh, first in human. Okay.
Right. When you are introducing an entirely new robotic system into the beating right atrium of a living person for the very first time, you do not want a fast global rollout. You need an intensely controlled, highly monitored environment with your absolute best clinical and engineering minds in a single room.
Ah, so it is a strict proof of concept. They aren't trying to cure millions today. They're just trying to prove that the robotic arm can safely perform the choreography in 10 consecutive patients.
The primary goal is demonstrating safety and feasibility. Once the protocol is perfected and the robotic system proves it can drastically reduce operator fatigue and minimize radiation exposure by allowing the surgeon to operate from a shielded console instead of standing over the table, the procedure becomes reproducible.
So you no longer need an interventional cardiologist with 20 years of hyper-specialized manual dexterity to pull off the surgery. You just need a trained operator and the robotic system.
Exactly. It democratizes the cure. It means this life-saving valve replacement could eventually be performed safely at standard regional medical centers worldwide, rather than being restricted to the top 1% of global surgical labs.
Wow.
We saw this exact evolution play out with robotic prostatectomies and robotic mitral valve repairs. It begins with 10 patients meticulously monitored in a single room, and within a decade, it becomes the global standard of care.
So what does this all mean? We've looked at a hollowed-out viral delivery truck carrying genetic blueprints, a biochemical barcode tracking a replacement enzyme straight into a muscle cell's lysosome, and a robotic catheter navigating the chaotic blood vessels of a beating human heart.
When you synthesize the data across all three of these incredibly diverse trials, the unifying theme is the mastery of delivery. Modern medicine has largely isolated the root causes of these devastating diseases.
Right, we know it's broken.
We know exactly which gene is mutated, we know the precise enzyme that is missing, and we understand the anatomical failure of the leaky valve. The frontier we are conquering today is overcoming the intense biological and physical barriers of getting the cure exactly where it needs to go without, causing collateral damage to the rest of the body.
And if you're listening, that is exactly why you should care deeply about the science unfolding in these specific trials. The ultra-rare gene therapy trial for young children or the 10-patient robotic study in Hong Kong might seem isolated, but they are actively laying the foundation for the medical infrastructure of the future.
They really are.
It is the slow, highly methodical process of taking diagnoses that were once labeled fatal or inoperable and transforming them into manageable conditions.
It represents a monumental shift in our capabilities. But as our ability to physically and biochemically deliver these treatments approaches near-perfect precision as we master the robotic arms and the mannose-6-phosphate barcodes, we are moving toward an entirely new challenge. Well, the next great frontier in medicine will likely no longer be about identifying the disease or navigating to the target. The next frontier will be predicting and controlling our own immune system's complex systemic reactions to the miraculous cures we are introducing.
Wow. So once we finally build the perfect master key to open the door, we still have to ensure the biological alarm system inside doesn't panic and burn the entire house down.
Exactly.
We're not just picking the locks on these untreatable diseases anymore. We are completely redesigning the hinges, we are redrawing the blueprints, and we are proving, trial by trial, that no door stays locked forever.